As of now, invasive species are seen as one of the greatest threats to biodiversity. In order for an invasive species to be a successful one, it needs genetic adaptations that allow for it to thrive in a non-native environment. In California, Cicimbrium aereo is a non-native plant species that grows throughout most of the southern half of the state. One of the areas that Cicimbrium aereo can grow in is part of a small serpentine region in the San Rafael Mountains that is inhabited by the endemic species Calanthus and plexicalis. Although their area of growth overlaps, there is no indication that the species interact with one another. The aim of this study was to use genomic analysis, along with what is already known about preferred soil composition and phenology, to find differences between the two species that may inhibit competition.
To answer these questions, we sorted through tandemly duplicated genes of Calanthus and plexicalis and Cicimbrium aereo, allowing us to organize them based on unique and shared genes. The genes were then further sorted based on the relevance of their function to our main research goals, such as those genes involved in heavy metal transport and stress response. We chose to filter the genes this way in order to help us understand how each plant responds to environmental stresses because, as shown by the map, Cicimbrium aereo inhabits a much wider range in California than Calanthus and plexicalis, despite the fact that Calanthus and plexicalis' ability to survive in serpentine soils might lead one to believe that it could grow anywhere. To further explore this idea, we then looked at the shared genes of interest between the two species to compare the number of copies each possessed and compiled it into the table shown.
After analyzing our results, we found that there was no significant difference in the number of genes for harsh environmental conditions that the two species shared. As you can see, they have a similar number of these genes that we selected for. So, as of now, we believe that the lack of competition between the two species may be due to other habitat preferences of the species rather than soil composition or environmental stresses. These factors can include the bloom period and preferred elevation. However, soil composition cannot be ruled out entirely. This data opens a lot of doors to future study potential. Some other tests that can be performed on the data include looking at the genes unique to each species and seeing if there's a difference in the number of genes there. There are also many more controlled experiments that can be done, such as attempting to grow Cicimbrium aereo in serpentine soil or placing the plants close to each other and seeing the effects of direct competition. Testing Cicimbrium aereo from different locales may also provide different genetic results. If we compared a Cicimbrium from a drier, warmer climate and one from a colder, more temperate climate, we may find that one of those Cicimbrium is better adapted to compete in the Calanthus' habitat. We wanted to thank Dr. Edger for supporting us throughout this process, and thank you so much for watching.